Good morning, everyone. Welcome to our quarterly vision webinar. We are excited to have you here today. Just a couple of housekeeping things as we get going. All participant lines are currently muted and all of your videos are turned off. So once again, welcome. We're excited that you are joining us for this quarterly vision webinar that's sponsored by Johnson & Johnson. My name is Bailey Bostelman, and I am the Regional Field Director here at the Foundation, and we're excited to be with you all today. So before we get started, I'd like to briefly review a few details for this call. Currently, all participant lines are muted and without video. Please be aware that all controls are at the bottom of the Zoom window. This control bar may collapse when it's not in use. If you would prefer to prevent these controls from auto hiding, you can use the following keyboard shortcuts to change your Always Show Meeting Controls option. If you're using a Windows computer, you can press the Alt key, and if you are using a Mac keyboard, press Command and backslash at the same time. This presentation is also being recorded and will be available with closed captioning. To activate closed captioning, please select Show Captions at the bottom of the screen on the Zoom toolbar. Today's webinar is sponsored by Johnson & Johnson and will feature the experts shaping the next generation of eye care. Today, our expert speakers will share information about age-related macular degeneration. From diagnosis, treatments, and patient perspectives, our valued speakers will update you on the latest in this space. At the end of the presentation, we will open up the floor for more Q&A from our panelists. So whether you are a patient, caregiver, or healthcare professional, we hope this webinar provides valuable insights. Before we jump into today's speakers, I would like to hand it over to our Senior Director of Legacy Giving, Darcy Meadows, to share more on how you can make a lasting impact. Darcy, the floor is yours.
Good Saturday, everyone. And I am the Senior Director of Legacy Fighting Blindness. I'm going to take just 3 minutes of your time to share about the importance of legacy giving at the Foundation, and then I'll turn it back to the wonderful scientists and clinicians you've tuned in to hear. I want to start by asking a simple question. What if you could change the future of the Foundation forever without writing a check today? Legacy giving, or giving from your estate plan, allows you to make a lasting impact on the Foundation. In fact, it's one of the most meaningful and sustainable ways you can support our mission. Scientific breakthroughs do not happen overnight. They require bold ideas, years of discovery, careful clinical trials, and sustained funding. The therapies that are restoring vision today began as research projects decades ago, and the cures we are working toward right now, they will depend on investments we make today and tomorrow and long after we are gone. That is why legacy support is so powerful. When someone includes our organization in their estate plans, they are doing something extraordinary. They are saying, I believe in a future without blindness. A legacy gift, number one, costs nothing today, and number two, is completely flexible and can be changed at any time. Yet it allows the Foundation to fund high-risk, high-reward science, support emerging researchers, sustain long-term clinical trials, and move discoveries from the lab to the patient. Legacy giving is not about wealth, it's about values. Legacy support ensures our progress never pauses. Imagine a person diagnosed today with a retinal disease. Many people on this call know a person affected, or maybe you yourself are affected. Now imagine that 10 to 20 years from now, because of research we funded, that person has access to treatments or even a prevention for age-related macular degeneration. Legacy donors may never meet the person whose sight is saved, but their generosity makes that breakthrough possible. And there is something deeply meaningful about knowing that your values, your belief in science and hope will continue working in the world beyond your lifetime. Today, I invite you to consider becoming part of our legacy society. You can include us in your will with a specific amount, a percentage of your estate, or it can be as simple as naming the Foundation as a beneficiary of a retirement account or life insurance policy. All commitments of any size are meaningful and deeply appreciated. Many legacy gifts are simple to arrange and do not affect your finances today. What they do affect is the future of vision research. As community members and close allies of the foundation, I'm asking you to consider what a legacy commitment to the foundation could mean to you and your family. Because when you really think about it, when you choose to place any charitable organization in your estate plans, you are elevating that organization to the level of family. Once you've made this decision, we encourage you to tell us about it, not only so we can plan for our future, but so we can thank you for your future generosity during your lifetime. The Legacy Giving team is here to help you. Please don't hesitate to reach out to me or my colleagues with inquiries. While we won't be taking any questions about this topic today, my email is on the screen, dmeadows@fightingblindness.org, and will be provided in the chat and follow-up materials. Thank you, and I'll field it back to Bailey for the rest of the seminar.
Great. Thanks so much, Darcy, for that information. I think legacy giving, like you said, is a great way to make that lasting impact. So now we're going to go ahead and switch gears a little bit about and looking a little bit about to the research that is being done and that we can help fund through those gifts. So we have 4 phenomenal presenters today who are going to share various topics related to age-related macular degeneration. And so first, we're going to hand it over to our first speaker, Dr. Patricia Diaz-Moore. So, Dr. Diaz-Moore, I'm happy to hand things over to you.
Okay, thank you very much. Actually, my affiliation on that slide was not right. So here's my current affiliate. Oh, it's never there. So this is my affiliation right now. And I just want to say that I'm going to share this. And you can tell me when it's shared. Is that good?
We are not seeing anything yet.
Really? Oh, huh. Share. Share. I have,
And there are you hitting the green button in the middle of the bottom of the Zoom toolbar?
No. Should I be?
Yes, hit the green button and then it should let all of us see your slides.
Wait a minute. I don't see the green button in the middle of the Zoom toolbar. I'm sorry, I Zoom all the time for a living, basically. So this is surprising. Anybody else who's on Mac have a suggestion for me?
Maybe you should log out and log back in, Pat, because I do see the share button at the bottom of my toolbar on Zoom, and I'm using
And
You
that.
do?
Yeah, I
if
do.
it is easier for you, if you want to throw the slide deck over to me via email, I'm happy to share on your behalf if to make it
Okay, I'll do that. And in the meantime, I'll talk anyway, which I'm capable
perfect.
of doing. So let me see if I can multitask by finding Bailey's email and simultaneously, okay, found her. So what my goal is going to be today is not so much about my own research, but what I really want to be able to do is to make sure that everybody out there who are suffering from some vision disorders or the pathology that we're talking about, AMD, and their caregivers have a good idea of really what the disease is all about. I mean, many of you may have been it may have been explained to you well by your clinicians, but many clinicians don't have time to get into the weeds. And I have some nice cartoons that will help you. I'm not going to be at a very high level, so not to worry about it. I'm forwarding these now to Bailey. Let's see if she gets them. So my goal is going to be, I'm going to show you like a picture of the retina. I'm going to give you a preview right now. I have cartoons about what I want to distinguish for you is the difference between wet AMD and dry AMD. I'm going to show you how they, from a cartoon point of view, how they actually form. Did you get it, Bailey?
Not yet.
Okay. How they form, and why they become a problem for vision. In the case of wet AMD, I'm going to talk about, the major treatment, the basis of the major treatment right now. Give you a little idea of how basic research which both Bailey and Darcy were talking about, have contributed to that in a major way. I doubt that we would have those treatments right now if basic research hadn't been done on those. Did you get it yet? It says it was sent, I think. Let me see.
Yes, and I
Yeah,
just
it says
checked
it
my
was sent.
spam filter to make sure it didn't get caught in
Yeah,
that.
it says it was sent, so, and it's not that big. So I'm going to tell you a little bit about how the basic science was done. Very little, just the concepts behind it, and how it has turned out to be a useful approach for a number of eye diseases. And then I'll switch to talk a little bit about, and I'll show you the difference between wet AMD and dry AMD, again, from a cartoon point of view, and why the current treatment for wet AMD is not what you want to be using for dry AMD. I'll mention, did you get it yet?
Not yet. I'm not sure where.
Yes, it shows sent. Um, it shows it's sent. I do not see a green button at the bottom of my Zoom. Sorry.
Now it just came through. Perfect.
Yay!
Yes,
Okay,
we did
I've already
it.
given you the talk. Now you'll get the cartoons to go with it. Um, and I'm sorry about that. I have to see why this didn't work for future reference. And when we were chit-chatting, I should have practiced and to make sure I could open it for you all. So,
No
are you
worries.
there yet?
Yes, I have it and I am sharing now.
Yay! Okay, I didn't. I'll talk a little bit faster. Perfect. Perfect, perfect, perfect. So this is me. I'm actually a PhD, meaning I do not treat patients. I have been working in the area of ophthalmology for a long time, so I feel like I know a lot about it, but I'm not somebody who treats patients. So any of those questions are going to go to my esteemed colleagues here. This is where I am right now, and one of my major areas of research right now is being interested in dry AMD, which I'll talk about in a minute. So do I, oh, I have to just say, next slide. So these slides are animated, so I really want you to go right from the top, the biggest structure we're talking about, the eye, down to, you'll see that's where the eye fits in. Next slide. And then that's going to get blown up here. So you've seen this, maybe or maybe not, you've seen this picture. This is if you took an eyeball and cut it down the middle like a slice of bread. And on the front of it, you see, I don't know if you can see my pointer, but in the front of it where it says lens is the front of the eye that faces out towards the environment. And any of you who, Kapil, or anybody who wants to jump in with a comment or a question, please do. That's where the light comes in. And the light comes in and goes to the back of the eye, which is the retina. This is the optic nerve. This is like the nerve through which your retina communicates with your brain. Okay, next one, we're going to turn the retina on its side. Now, this is a cartoon. Let me point out to you that this structure of the retina is literally as thin as a piece of tissue paper. You're getting the impression that it's much thicker because I'm showing you this cartoon, but it's literally the thinness of a piece of tissue paper, yet it has all of these important layers in them. Next one. So this is it blown up. And again, we're going to drill down to where the disease process happens. So this up here is if the light were coming in, it would come in through the top called the vitreous. So it passes through the cornea, passes through this gel called the vitreous, and comes upon the retina. So that's the vitreous. And then the layers that we're most interested today are here at the back. It's the choroid at the bottom that you see, that's a blood supply. That's a blood supply that's responsible for nourishing everything above it to about the middle of the retina. So it's a very important source of oxygen and nutrients. Right above the choroid, there's a blue line that's called Brooks membrane. It's basically like another piece of cellophane on which cells can sit. And right above that is a very important cell type. I've called it RPE. It's retinal. Okay, so it's in the retina. It's pigmented. It's not shown here, but it is pigmented, and it's an epithelium. So it's clever. It's well named. So retinal pigment epithelium, and I'm going to be referring to that. And this is where we have some problems back here in macular degeneration. I'll refer briefly to what happens up here, just so you know how to distinguish between them or how to think about them. So that's what we're going to be talking about is this important complex back here pigment epithelium sitting on Brooks membrane, sitting above choroid. Very important supply of oxygen and nutrients. Next one. Okay, you can go. Yep, I already said that. And then I just want to be, again, very big picture. Your vascular system goes all the way from your heart, which is part of your vascular system, all the way down. Next two slides, you can just animate them down to the tiniest capillary. So this is a very cool picture. I'm sure you've seen this from some of this uh, um, exhibits that have gone around. But here you can see this big blood vessel. And then see how red this is? That's because there's so many capillaries, these little capillaries on your fingertips, because that's where all the sensation happens. And those nerves there need that nutrition. So think about it as, a very localized source of nutrition for this very highly innervated part. And then this is just a cartoon, but it shows it's basically big enough for one red cell to go through. And that's what we call the microvasculature or capillaries. And that really causes us, it's critical. It's a major part of the vascular system, but it also can be a problem, as we'll hear. Next. So we're going to talk about and again, we're talking about a very, for lack of a better word, stylized or cartoon version. This is a picture of the thickest part of the retina. I'll refer to you in some pictures later. The part called the macula, which is a very tiny part of the whole retina. We'll have a picture of that later, but we're back here. So what happens in macular degeneration next is you start to accumulate, I'm sure many of you have heard of it, called drusen. It's basically debris, and we're not going to go into where that comes from right now, but this debris does several things. It gets in the way of the nutrients coming up from here, right? Because it's basically a hill in the way. And then the cells above it aren't happy about it. They like being on their old Brooks membrane. They do not like this pile of debris in their neighborhood. And so they start to get pretty sick. Next slide. And you can lose those cells. They're not always lost. Sometimes can get sick, but then the cells from underneath here, these capillaries, start to grow through. They're not supposed to. They're supposed to stay in their own neighborhood. They're not supposed to be migrating either under these cells or into this space. And the unusual thing about new blood vessels like this is that they're leaky. These blood vessels down here, oxygen gets through and nutrients get through, but these actually can lead fluid. Like when you get a mosquito bite and you see a big puff there, that's from fluid that's leaking out of your capillaries. That fluid is what turns out to be really a problematic when it comes to vision. So next one. So this is what that might look like. Those blood vessels can leak blood or fluid under the retina, and that's a problem because it disrupts. Remember how fine-tuned I showed you that architecture was. You start introducing fluid into that space, you're very much messing up with the physics and the biophysics of that very finely assembled retina, and that becomes a problem. So the next one was, how do you get rid of that? And people have been trying for a long time. I'm going to skip through this for now. Everybody knows it's a very common problem. I'm going to spend three slides telling you about how basic science has led to contributions in treatment of wet AMD. So this was actually my mentor, Judah Folkman, and he was interested in tumors. And next slide, I'll show you, came up with the hypothesis that if a tumor, because it's big, a tumor is big like a big piece of tissue, it can get big. If it didn't have a blood supply, next, just animate it. Once you give it a blood supply, it can grow. But if you could prevent a blood supply from growing into a tumor, you could prevent tumor growth. And that was his very big interest from the 70s. And much of his career was spent on trying to figure out how to stop these blood vessels from growing. Well, he also realized that that translates very importantly to the eye. What did I just show you before? Blood vessels growing where we didn't want them. So a lot of work was done from him and many other people. I can't even begin to mention the number of people. The next slide was they identified a molecule which you have all heard of. So this is a healthy blood vessel. Remember, I told you it's not leaky. Here's the tube that the red cells go through. You don't really need to know, but these are called endothelial cells and pericytes. But that's what it takes to make a healthy blood vessel. But if you have growing blood vessels, you can animate it. Now hit it again. Those that boundary, that barrier breaks, and these cells migrate out to make new ones. Where do you think those new blood vessels are coming from? They're coming from pre-existing blood vessels, and they're stimulated by what? This molecule, other molecules, but in this case, for our purposes, very important. Vascular, endothelial, that's what these are. Growth factor. Again, not badly named. We're making the vascular endothelial cells grow. And it was shown through a variety of experiments, both in tumors, but most importantly for our purposes in the eye, that this was a key factor in making blood vessels both grow and be leaky. You see how they're leaky here? There's a space. This is not as tight as this. They're not only growing, but fluid is leaking out. And so it was determined, you can hit it again. It was determined with all kinds of anti-VEGFs that are out right now, I named two here, that if you put an antibody to those, you could block the activity of this growth factor that was causing both growth and permeability and stop the growth and permeability. Next slide. So for our purposes, what's so important about that is, next one. This is what many of you have either had or heard your loved ones talk about getting an OCT. Basically, it's a spectacular form of imaging that non-invasively allows clinicians to look to image the eye and get basically a cross section of the eye in real time without damaging the eye in any way. So this is the retina that we talked about, but there's also this very specialized area which we haven't referred to yet called the macula. That may sound familiar, macular degeneration. And the reason that it's called macular degeneration is when the disease process starts to encroach on this little area, it's 1% of the whole retina, then you really notice central vision issues. Okay. And there's specializations here you can see. So there's a lot of research going on about why is the retina so vulnerable in that position. This area, 1% of the retina, is responsible for your central vision. When you disrupt the morphology and function here, you have a central vision defect. You can still have all of this out here functional. Peripherally, it can be functional, but you're going to have a dramatic deficit centrally. Next slide. I only have a couple more to go and I'm almost done. This is just a dramatic picture that I got from the internet to show you why anti-VEGF works. The picture that I just showed you of the OCT, remember, it was flat. When you have blood vessels growing in the wrong place and they're leaking fluid, you have this big hump of fluid here, right? I showed you those vessels were leaking into a space where they shouldn't be in this fluid. For many people, if they receive an anti-VEGF injection, which I told you is going to block the effects of VEGF, that fluid will eventually get absorbed and you can have a flattened retina. Now, it's not perfect, right? I showed you. Remember, this looked significantly different. You have a little bit of a suggestion of where the macula was, but still, it's way better than this. And this is associated with improved vision. You can actually get improved vision from this point to this point. So the next one. This is just a quick point to, because I'm sure you also have friends or family who might have had proliferative diabetic retinopathy. I'm just going to show you in two slides what the difference is. In wet AMD, the problem is here. In diabetic retinopathy, it's up here. So you can hit the next one. Blood vessels grow in an unwanted fashion from here, and they also leak. But fortunately, they're also vulnerable to anti-VEGF. So it's turned out that this therapeutic, because this is a VEGF-driven process also. So same basic pathology, blood vessels where you don't want them, but in two very different parts of the retina. Next one. And yeah, that's what you can see, these all new blood vessels in here, little fronds. You can hit them twice and you'll see that's just pointing to the new blood vessels, again, where they're not supposed to be. And I think I have three more slides about dry AMD because I want you to know the difference there. So dry macular degeneration, again, this is very stylized, meaning it's sort of averaged out. We're not talking highly scientifically, but we're still, next slide, we're in the same problem. You can have debris forming here, okay? And again, these cells are not happy. And what can happen is, next slide, I mean, next just animation, these cells will die. But instead of blood vessels coming up in this case, what happens is these cells are very communicate a lot with these little blood vessels down here. So these are the big feeding vessels. I'll show you that in a minute. These are the little capillaries where the exchange of oxygen and nutrients happens. But this blood supply needs these cells to continue existing. So when these our retinal pigment epithelial cells go away, what eventually happens, next one, Bailey, is the blood vessels down here go away, and these photoreceptors, where all the vision is happening, go away because they need these RPE in this blood supply. So hit it again, and Bailey just, and then basically go back. Sorry, that gives you basically a blind spot. Now, if that blind spot is small and in your periphery, you might not even notice it, or you'd see it on, you know, uh, what's the test I do all the time? Where you're looking for blind spots. You could pick it up if you look very closely, but if this is in the macula, then you have a problem, right? Next slide. And when it gets very, very advanced, it's called geographic atrophy. And what I'm going to draw your attention to here, so this is a big area where the RPE cells have died, right? All the pigment is gone, right? Because this is some of what you're seeing is pigment. And not only are the RPE gone, the little vessels underneath, these are big vessels now. Why are these big vessels? Because the little ones have died and they're gone. And this tends to expand from the edges out. Last slide, I believe. Yep. So what do we know? For dry AMD, you can take lutein means you can hit it twice. You can eat your leafy greens, and you can take your AREDS vitamins. Um, I might have done that. Did I do the next one? Go ahead. But for geographic atrophy, which is the very, this is before you actually develop lesions, as I showed you, there are two treatments, and may have heard of them, but they have not been shown to preserve vision. So there's some controversy about how useful they actually are. So that's a major area of unmet need in biology or in vision research right now, what a lot of people are looking at. So I'll stop there. And that's it. Thank you for your attention and sorry for the technical difficulties.
Wonderful. Thank you so much. And no worries at all. We figured it out. And we'll open it up to some more questions here at the end. Until then, I'm going to go ahead and pass it over to Dr. Zarbin to get us going on some other questions.
Thanks very much, and thank you, Pat, for that prelude. My goal today is really to answer your questions, so I'm just going to make some very brief comments about photobiomodulation therapy and anti-complement therapy for geographic atrophy. And then, if it's appropriate, I'll show some slides. The idea of photobiomodulation therapy is you illuminate the eye with long wavelength light. And your goal is to try to stimulate a subcellular organelle called the mitochondria. And the mitochondria, as you probably know, generate energy, ATP. And it turns out that one of the elements in the mitochondria called cytochrome C, when it's illuminated with long wavelength light, say red wavelength or infrared, actually will increase ATP production. And the idea was that maybe this would help people with the intermediate stage of macular degeneration. So this is a stage of the disease that is present before you get to abnormal blood vessel growth, before you get to geographic atrophy. And a study was done uh, that showed some benefit. I think there are some concerns about the way that study was done, but the FDA has approved the use of this laser device. And And so you may have heard of it, you may have questions about it, you may be interested in what my reservations are about it. Happy to share all that with you if you want to. As far as the geographic atrophy, this is the more advanced form of the dry AMD, no abnormal blood vessels. There are two drugs, as Dr. Diamore mentioned. They both work off the same principle. So let me take a minute to explain the principle. Age-related macular degeneration, like many age-related diseases, is characterized by chronic inflammation. And Alzheimer's disease, for example, is characterized by chronic inflammation. And it turns out that um, there's a certain class of molecules called complement factors that are very important in mediating chronic inflammatory diseases. And this ranges from everything from rheumatoid arthritis to geographic atrophy. So the treatments for geographic atrophy that have been approved by the FDA are both complement inhibitors. Uh, One is called pegcetacoplan, and one is called avacincaptad. Now, they're both complement inhibitors, but they're not identical. They're not identical biochemically, and they're not identical biologically because this complement pathway of inflammation has different checkpoints in it, and these two molecules act at different points in the checkpoint pathway. The studies that demonstrated that led to the approval of these two geographic atrophy treatments were both very well conducted in the sense that they're both multi-center, randomized, prospective clinical trials. They were conducted during COVID, which led to some technical difficulties, but amazingly, they both showed pretty much the same thing, which is that if you use these inhibitors of complement activation, you can reduce the rate of progression of geographic atrophy by somewhere in the range of 15 to 20% over a 2-year period. And of course, this is a funny situation to be in if you're a patient, because in order these drugs wear off, so you have to get repeated injections of the medicine in your eye every, say, 4 to 8 weeks. And if the medicine's working, you won't know anything because it's not making you see better. In fact, what it's doing is slowing down the rate at which you lose vision. So maybe one way you could think about that is if you're driving toward a brick wall at 100 miles an hour, and if I slow down your car to 80 miles an hour, you're still going to hit the brick wall, but you're just going to hit it at a little bit later in time. And so the idea is to try to preserve the central vision a little bit longer by using these medications. And of course, like everything in life, these medications can have complications. Both of them, interestingly enough, and can be associated with an increased risk of developing abnormal blood vessel growth in the eye, the choroidal new vessels that Dr. Diamore mentioned to you. I'm going to say a little bit more about that in a minute. The other complication that seems to be pretty much associated with the pegcetacoplan is the risk of very severe inflammation of the blood vessels. It's very uncommon. It probably happens to 1 in 4,000 patients, and it seems to only happen after the first injection. So if you get the medicine and it hasn't happened by the second or third injection, it doesn't seem that it's ever going to happen. Now, let me back up about these abnormal blood vessels. We, you know, the abnormal blood vessels, as Dr. Diamore pointed out, generally cause severe visual loss, but they grow for a reason. They grow in response to inadequate oxygenation. And if the blood vessels didn't leak, they'd actually be therapeutic. And it turns out that not all of these abnormal choroidal vessels do leak. And in fact, when they don't leak, they prevent the spread of geographic atrophy. So the development of the abnormal blood vessels in the patients who receive the anti-complement therapy is something that has to be managed in a very individual way by the doctor taking care of you. If the vessels aren't leaking, probably they should just be watched because they're doing good. If they're leaking, then you'll need the treatment for the wet form of the disease in order to control the leakage. So those are the general comments that I want to make, and I'm happy to show slides if you want, but I mostly want to be sure I answer any questions that people in the audience have.
So there's been a couple questions that have popped in from the chat. Two of them are around the laser treatment that you mentioned. What are your reservations? And somebody who experienced this treatment over 20 years ago mentioned that there was also some good cells that potentially was damaged. So is this still the case? What are those reservations that you have?
Uh, Let me first make the distinction between the treatment, laser treatment 20 years ago and this particular treatment. The laser treatment 20 years ago was a much higher energy delivery, the goal of which was to make the drusen that Dr. Diamore pointed out go away. And in fact, that 20 years ago laser treatment did make the drusen go away, but it also induced the growth of abnormal blood vessels. Now, this laser treatment, the photobiomodulation therapy, is a much lower power, so much less intense energy. But interestingly enough, there may be some evidence, it's weak evidence, that it increases the risk of choroidal neovascularization. So this will bring me to my reservations, my concerns about the study. Now, the first thing you have to understand is that this laser treatment is fundamentally different from a drug treatment. Drug treatments, when they're being reviewed by the FDA, generally require something called a parallel group clinical trial, which means two clinical trials, each of which has a control group. Because this laser treatment is a device, that there's a different paradigm for regulatory approval. You don't need two trials, you can do one trial. The second difference, because this is a device, is the threshold for determining approval is actually different. So in a drug trial, you generally need to show improvement of 15 or more letters and a risk of visual loss no more than 5 letters for the majority of the patients. So on average, let's say. That's not true for the device trials. You just need to show an improvement of I think it's 5 letters and no visual loss. So the barrier for approval is fundamentally different because this is a device and not a drug treatment. So that's one thing that we need to kind of keep very clearly in our head. Yeah, 5 letter loss is you don't want to have um, uh, more than you need a 5 or more letter improvement over 24 months, and you can have evidence of harm. Now, this trial was a small trial as far as this goes. There were 100 subjects enrolled. Sometimes both eyes of the patient were enrolled, so there were 148 eyes. So the first problem was there was a lot of dropout in the study. By the end of 2 years, 22% of the patients had dropped out. That can lead to concerns about whether there's a bias in the analysis of the data at the end of the study, whether there's what's called external validity of the study result. In other words, will you get the same result in the real world as you got in the trial? And I think that from the perspective of the statistical analysis, that those issues were recognized and handled appropriately. But if we just look at the actual results themselves, the patients by month 21 who received the treatment had about a 6 letter gain in improvement, and the patients who were in the control group got a 2 letter gain. Now, a 4 letter difference in the treatment versus the controls is something that's actually at the very edge of what can be reproducibly measured in clinical trials. And generally, in a small clinical trial, that's something that we'd be a little concerned about. And in fact, if you look at the variation in the visual outcomes, it's rather large. So the first question, maybe I could rephrase it, is the clinical benefit that was measured clinically meaningful for most patients? Very related to that is if you look inside the trial and you look at the visual benefit for people that entered with different levels of visual acuity, here's what you see. For people that entered with vision of 20/40 or better, there was no visual benefit. It was the people who had vision worse than 20/40 that got the visual benefit. Now, when you have this intermediate stage of age-related macular degeneration, the overwhelming number of people have vision better than 20/40. So the overwhelming number of people have vision that wouldn't even allow them to be enrolled in the trial. And based on the trial result itself, probably wouldn't derive any visual benefit from it. Uh, this is a problem, in my opinion. I could also go into another detail, which is that if the patients had intermediate AMD, you would expect them all to have vision of 20/40 or better, but yet they had patients who had vision of 20/70 who were judged to have intermediate AD. So it's a little I'm a little bit concerned about exactly how the enrollment criteria were applied in the real world to enroll the patients. Another concern about the results has to do with the claim that this actually reduces the rate of development of geographic atrophy. Well, um, one of the claims is based on the fact that, geographic atrophy doesn't just happen, like a thunderstorm. It's sunny and all of a sudden there's a dark cloud and lightning and rain. It's something that happens gradually. And in the early stages of it's termed incomplete RPE and outer retinal atrophy. And they found that the likelihood of developing incomplete RPE and outer retinal atrophy was reduced by the photobiomodulation therapy compared to the controls. And this sort of creates a kind of a logical argument to say, so we're probably going to reduce the risk of getting geographic atrophy. But in fact, this early stage of degeneration can reverse. So not everyone that gets it goes on to develop geographic atrophy. So that's a claim that remains to be validated. If you actually look at the actual development of geographic atrophy, the number of patients enrolled in the trial that did so is so small. There was a difference numerically between the treatment and the control, but the number of patients that actually had this was so small, it would be just unreasonable to make the claim that you can prevent the development of geographic atrophy based on these data, my opinion, um, uh, you know, alone. Um, so the final thing is the issue of, are there any complications with the treatment? Now, I don't want to, I want to be very careful that you understand what I'm trying to communicate. If you look at the number of patients that developed choroidal neovascularization, there was actually more patients who developed it who received the treatment than who didn't receive the treatment. I am not asserting that this treatment causes choroidal neovascularization. I'm asserting something a little more subtle. If you go back inside the trial and look at and try to answer the question, why did more patients who got the treatment develop choroidal new vessels compared to the ones who didn't get the treatment? What you discover is that there were more patients in the trial who had their fellow eye with choroidal neovascularization among the treatment group. And if you have choroidal neovascularization in one eye, you're more likely to get it in the other eye. And you could say, well, there's a control group. But in fact, the control group wasn't balanced in that way. So this imbalance in the incidence of choroidal neovascularization actually means to me that the treatment and control groups were actually not balanced in their risks for developing the late complications of AMD. It doesn't necessarily mean, and I kind of doubt that it means, that the treatment causes choroidal neovascularization. These are questions that I think can only be answered with more data. There's one other little detail that I think is relevant for the real world, and it has to do with the way the treatment is administered. So the way you receive it is you get 9 treatments over a period of 3 to 5 weeks. And this 9 treatments are repeated every 4 months, which means in a year, you're going to have 36 office visits. Are 36 office visits worth 4 letters of visual improvement? Of course, I would never, ever want me to answer that question for you, and I don't want the government to answer that question for you, but I want you to answer that question for you.
So, Marco, there's a question that might be worth answering now, but what do you mean by 4 or 6 letters? What does that mean to a patient, or how is that determined?
Well, so there are different ways we can measure the visual acuity. And of course, as you all probably know, we use these visual acuity charts, and there are all kinds of different charts. And when you're doing a clinical trial, in ophthalmology, one of the important things to get really accurately right is what the patient's visual acuity is. So it turned out that there was a chart that was developed precisely for this objective, for the reproducible, accurate measurement of visual acuity. It's called a Bailey-Lovie chart. And it has the same number of letters on each line, and it has the same spacing of the letters on each line. And there are 5 letters on each line. And it turns out that when you have this kind of very strictly organized approach to measuring vision, the minimum change in vision that you can measure reproducibly is around 4 letters under optimal circumstances. Okay, that means the same illumination, the same person doing the refraction, the patient blinded to which eye would they get the treatment or not get the treatment. And unfortunately, the worse your vision is, the a more unreliable the 4 letter difference is. So the 4 letter difference is most reliable when you have, if you're going from 2040 to 2030, let's say. But if you're going from 2070 to 2060, it's really not very reliable at all. I hope I've answered your question.
Great.
That phenomenon, by the way, for those of you who are familiar with statistics, is known as heteroscedasticity, which is that the variation is variable depending on where you are on the x-axis.
So, looking at one of the questions in here about this laser treatment is what is the name of this treatment again, and this versus using something like Irvirze?
Using AREDS, the name of the treatment is photobiomodulation therapy, PBM, and it is not an alternative to using the AREDS therapy. The AREDS therapy is a pill that you take in the morning and in the evening. And to be clear, the AREDS therapy reduces in the prospective, multi-center, randomized clinical trials that have been done, the AREDS therapy reduces the risk of moderate visual loss. That's 3 lines of vision on that eye chart I was talking about, by about 29%, 19%, sorry, over a 5-year period of follow-up. And it actually doesn't reduce the risk of visual loss for geographic atrophy. It reduces the risk of visual loss associated with choroidal neovascularization. In fact, someone may have asked this question, Bailey, so I'm going to answer it ahead of time. There was some evidence that was published, high-profile publication, indicating that the AREDS treatment itself might be as good as the anti-complement therapy. But in fact, and I want to disclose I'm a consultant for Apellis, the company that did the study I'm about to tell you about. One of the companies that markets one of these anti-complement therapies had a beautiful data set that they could look inside and actually see whether or not the AREDS therapy made any difference whatsoever in the rate of progression of geographic atrophy if you didn't get the complement therapy and if you did get the complement therapy. And it made no difference whatsoever. So AREDS treatment, in my opinion, based on the analysis of randomized prospective clinical trial data with appropriate controls, is not a treatment that reduces the rate of progression of geographic atrophy.
Perfect. Thank you so much for that. And for the sake of time, we're going to go ahead and switch gears a little bit and hand it over to Dr. Bohardi, and we'll go ahead and address some more of these questions throughout. But thank you, Dr. Zarbin. That was fantastic information. We really appreciate it.
My pleasure, and thank you for inviting me to speak with you today. Bye-bye, everybody.
Bye, Michael. Thank you, Bailey, for inviting me to speak. And now I'm going to talk a little bit about, in the next few minutes, a new and upcoming treatment for macular degeneration, both for dry AMD and for wet AMD, which is a stem cell-based therapy. You heard from Dr. Diamore that in late stages of disease, the vision decline happens because retinal pigment epithelium cells start to die off, and as a result of RPE cells dying, the photoreceptor cells that are the light sensing cells of the eye die off, and that's the reason why vision declines. but multiple studies have shown that RPE cell loss precedes photoreceptor cell loss. And that led to this idea that what if we could transplant a piece of RPE in the area where the photoreceptors are still viable, and that transplant could then actually protect those photoreceptors from dying. And this, in fact, has been tested in clinical surgical approaches where patients on retinal pigment epithelium from the periphery of the same eye was transplanted in the macula, and it indeed, in some cases, preserved patients' vision for years to come. So then the question comes is, how do you make a retinal pigment epithelium? And in fact, you could also make a photoreceptor implant from stem cells. And I call this living drug because these cells, this drug is going to live in patients' eyes for the rest of their lives. Unlike any other small molecule drug, which clears out within hours to a few days, these cells stay. Hence, they interact with the eye, they interact with the environment. They would change, they might change or might not change, but they would interact with the environment. Hence, it is very important for us as we manufacture these stem cell based therapies to be very careful what we're designing, what we are making, and do all kind of quality controls before we transplant them into anybody's eyes or any other organ. So how do you make, why choose stem cells to make RPE cells or photoreceptors? Stem cells have this very unique two properties that first of all, it's a cell that can make multiple copies of itself. So you can grow it almost infinitely. And then a stem cell can also make all the different tissues of the body. And some examples are listed here. It can make neurons of the brain. It can make blood cells. It can make muscle cells. It can make liver cells. And obviously it can make cells of the eye that we're going to make here. Broadly speaking, there are three types of stem cells. Top two are have the quality that they can make any cell type of the body. First is embryonic stem cells, which are derived from a blastocyst. They're known for their ethical concerns and political controversy around them. But we don't have to worry about those controversies at this point because we don't have to use embryonic stem cells with the new discovery of what is called induced pluripotent stem cells, iPS cells. These cells have exactly the same property as embryonic stem cells, as in they can make any cell type of the body. But the beauty is that you don't have to destroy an embryo to make these cells. You can make them from any adult. You can take a little bit of the skin, their blood, convert them into iPS cells, the induced pluripotent stem cells. And the second advantage that provides us that because you can make it from somebody's own cells, you can actually make their own tissue in a dish and then give it back to them. So that's autologous transplant. And the big advantage of that is then you don't have to immunosuppress the patients, and the transplant will likely survive for the rest of patient's life because it's their own tissue. It won't be rejected by their body. And the third stem cell type, which is routinely used in many conditions, which is the biggest example is blood stem cells, which is tissue specific stem cells. We all have blood stem cells. They renew our blood every 2 to 3 months. And then some other known examples for tissue specific stem cells are umbilical cord stem cells, mesenchymal stem cells. In fact, now there's a tissue specific stem cells for the eye as well, from the RPE as well, which is also in a clinical trial stage. And I'll talk about that in a minute. But before I go on, I do want to illustrate the power that the stem cell based therapies have. It's still all of these are still in development, but this one example really illustrates the power, and it's mainly using the third stem cell type, the tissue specific stem cell types. And this is a story of this little child, Toby, who's now 4 years old and is walking like any small child would walk at that age. But if it wasn't for the stem cell based therapy, Toby actually would be disabled by now and would not be able to walk. When Kobe's mother was pregnant with Toby, he was diagnosed with a defect in his spinal cord. And if he was born with a defect, he would be disabled and won't be able to walk. What doctors and scientists came together and they did is they developed a stem cell based placental stem cell based patch, which they delivered and transplanted onto Toby's spinal cord while he was inside his mother's tummy, and were able to repair his spinal cord. And when he was born almost like a normal child. So this technology has a huge potential, but as I again made the point that it is a living drug, and we have to do all kind of diligence to make sure that this drug is made made correctly, and all quality controls are done before it's delivered to patients. So now coming back to iPS cells, as I mentioned, you can make iPS cells, induced pluripotent stem cells, from any adult, any person. Take this skin cells or blood cells, you reprogram them with specific vectors in a dish, and then from those reprogrammed iPS cells, you can make blood cells, gut cells, muscle cells, and of course the eye cell. For instance, in our case, you can easily make from a patient blood cell, iPS cell, and then from an RPE cell. And here's an example of RPE cells made from patients on iPS cells. RPE cells, as if you remember Dr. Diamore's talk, she mentioned they are pigmented and they're epithelial in nature. Here you can see epithelial character is marked by the hexagonal morphology of these cells, and you can see they're fully pigmented. Hence, you can make mature, fully pigmented RPE cells from iPS cells. These cells are in many different clinical trials. Just in the interest of time, I'm going to illustrate a few of those. On top is an approach where you deliver these cells, RPE cells, as suspension into the area of GA or around the area of the GA injury or GA lesion. Two trials that are ongoing is one from Astellas Pharmaceuticals that uses embryonic stem cell derived RPE, and a new trial that has just started with Loxa Therapeutics, which actually uses adult RPE stem cells, not embryonic or pluripotent. At the bottom is a different approach of delivering these cells not as a suspension, but as a patch, which is now done by Regenerative Patch Technology in California, where they have RPE cells growing on a plastic patch, and they deliver the entire patch in the area of GA. The third approach is the work that we are doing at the National Eye Institute. In this case, we make iPSC-derived RPE, so from patients on iPS cells. So this is an autologous patch, and it goes on as a monolayer on a biodegradable scaffold, which is integrated into the back of the eye using the scaffold, but the scaffold degrades away within a month, so the cells would actually integrate using their own matrix around them, using their own Brooks membrane-like structure, if you may remember from Dr. Diamore's talk. So this is an ongoing phase 1/2A study at the NEI. So with that, I want to come to end of my talk, but before I close it, I want to make a point that just like any drug, any chemical, any antibody, any peptide, we heard two examples of peptide therapies. Stem cells are also like that. It takes years to put together a trial, FDA approval before going into patients, and then, of course, FDA approval at every step before market approval for a trial. But if you come across such cases where there are stem cell clinics, and I put the clinics under quote here, which often do not have FDA approval to transplant those stem cells in patients, be careful of those. There's a lot of unapproved and unproven stem cell therapies, stem cells being delivered to patients, and they're charging patients a lot of money to do that. And often, in many cases, they have actually harmed patients' vision. There's an example of a paper where 3 patients went blind after injections of wrong stem cells in their eye. You can get more information about approved versus unapproved stem cell therapies from the FDA website, from the NIH website, and from the International Stem Cell Society for Stem Cells Society. The bottom line is, if somebody wants you to pay out of pocket for a stem cell treatment, it is likely not approved. And currently, there is no FDA-approved commercially available stem cell-based treatment for the eye. All ongoing approaches are at a trial stage. So please be mindful of this, and please ask if the trial has gotten FDA approval before you enroll in it. With that, thank you very much for your attention. Happy to take questions.
Wonderful. Thank you so much for that. And I think that is a very, very good reminder when individuals are looking for potential treatments or trials, is that, you know, there's a lot out there on the market sometimes that looks promising, but really doing your research, talking to your doctor, talking to the scientists behind that, and like you said, if it's a promising one, likely they will not be charging you. It is something that is covered underneath the parameters of that clinical trial. So we are right about at the top of the hour at 1 o'clock here on the East Coast. So we've talked a lot about the eye and how the macula develops, how these drusen's developed. We've talked about some of the treatments that are out there, and then we've wrapped it up with some things about stem cell therapies, which are some promising, innovative treatments that could potentially help us all in the future. So before we open it up to some more questions, and Dr. Giamore, I see your hands up. So before I switch it over to Louis, well, I would love to answer
I
any
just want to
questions you have.
say one thing,
Yes,
which people probably already know, but that in addition, and besides aging, which is sort of hard to control, smoking is a major risk factor. So, there's many reasons to not smoke. This is a big one.
yes, I love that. That is a great reminder that there's a lot of factors that us as individuals can control that can potentially impact the progression of AMD. So that's a great reminder. Thank you for that. So I'm going to go ahead and switch it over to Louis. So while we're thinking about how we can impact the progression of these, let's also talk a little bit about what individuals can do today to help them live robust, healthy, happy life. So Louis is the patient that we have, and he's going to share a little bit about his experience. And you're currently muted, so if we can get you off mute here, we'll be good to go. Perfect.
Can you hear me? Okay.
Yes.
Oh, thanks for the invitation, and thanks for the opportunity of sharing my problem with everybody and what I have been doing. I'm glad that all the two things I have listened recently, I'm doing both. So the photobiomodulation is something I'm close to start, and I already have a new retina developed for me based on my stem cells. The problem is that still to implant is tough. I have recently they have changed the topology for this. What I have is an extensive macular atrophy, which I would name this in a simple way, which is AMD on steroids, which is more aggressive, is faster, and it's more complicated. The gene that I have not necessarily generate just a few drusens. I have hundreds of drusens, and vision I have today is around 2300 on one eye and 2600 on the other eye. So with that, let me just explain three topics that I think are relevant to mention. First one is how do you live and cope with your friends and family? Second one is about technology, and third one is about attitude. The first one, I think we all that suffer from the problem, we have a very hard time explaining what we have, because people say, you tell me that you don't see, and sometimes you see a coin dropping on the floor. So it's very hard to explain to people what we have. So what I have done, I went to a Home Depot. I bought a very simple safety glasses. I put a tape, a transparent tape in the middle, like a circle, and I did this to all my family. I make them use this with the circle in the middle, just the peripheral vision they would have. And I said, use this and walk around, see, eat, do everything. And that's what I said. And that helped a lot, because people understand what you see, why do you see, and why sometimes you see things that you're not supposed to see. So that has been very helpful. The other thing I have learned is sometimes, especially when in the middle, you try to hide it. So I would say that I've added an additional last name to my name, and I say I'm Luis Ferzin, and I have low vision. That's how I introduce myself, and that helps a lot, because we need to be humble, and that was very hard for me to be humble to explain what we have. The second component and point that I want to share with everybody is around technology. I think there's a lot of things going around, and I can share with you things here I have from the Iris Vision. I have a Ruby. I have an Orcam, and all sorts of things here. I can go on and on, because I'm a tech freak, and so I've tried everything. And if I have to give an advice to you, is don't get desperate. Sometimes we use technology, we buy too fast something and we're not going to use it. There are separate uses, there are different things that you can use for them, but try them a lot before buying something, because at the end of the day, uh, uh, you need to try for some time before you really know that's going to help you in what you need to do. And that's very easy to get a lot of devices on your table when you don't use most of them, or you just use for a few situations. What I also have learned a lot is that especially for older people, it's hard to engage with technology. But today, if you use a very simple iPhone and you connect CD with a ChatGPT, and you can see, can ask what medicine is this, what it's saying. There's so many things you only need to do with your voice. And I know people don't don't like to share or to engage with technology a lot because I think it's for younger people, they think. But things are much easier today. So the power you have with your phone in your hand is unbelievable. From apps like Seeing AI to linking your Siri with ChatGPT or Gemini, something very simple, and your life is going to change dramatically just by using that. Very simple things that you can do. The third component has to do a little bit with the attitude, and I always say that if you have, and I think all of us that are losing sight, we always have dreams that we dream that we're gonna get back to our old vision and see again in a better way. And I think that we all need to stick with these dreams, because dreams at the end of the day are the opposite of negative thinking. And if you have more dreams than negative thinking, is because you have hope, and hope is the most important thing regarding changing your attitude and how you do things. So I think this is a message and pass to everybody. Don't get negative. Chase your dreams, follow your dreams, and still have your dreams that we have just heard a lot of things that science is evolving very, very fast, and I'm sure that we'll be able to use that. If it's not the case, at least our next generation will probably benefit from that. But anyway, I think this is very important. And what I also do that's relevant for me is to stay fit. I think doing exercise is very important, because you need to stay fit and take care of your mental health, meditating or whatever you do to have a better mental health. So just I know that we're on top of the hour and past that, so I just want to share some of these experiences that I have done. So try to engage your friends and family to learn how you see it so they can understand. Second, be careful with the technology. Don't buy in too fast. Really understand if it's going to help you. And third one, work in having a positive attitude.
That is fantastic. Thank you so much for sharing those tips. I think those are key controllable pieces that everybody on this call can take advantage of and implement. And one question that came through the chat that you kind of alluded to, Louise, and then Dr. D'Amore, you mentioned it as well, about preventative measures for AMD. So whether it be diet, not smoking, I know that I've been told that a Mediterranean diet that has a lot of those, nutrients that would help. Are there any other pieces that you all would recommend that are just good lifestyle changes that could help individuals with AMD?
One of the recommendations I received from my doctor in which is in the Columbia Eye Institute, is basically exercise, not necessarily only aerobics, but to produce lactic acid as well. So that means doing weights and all this that can help as well. And as also we have heard that in the case of extensive macular atrophy, not necessarily AREDS is something that's going to happen to help.
Perfect. Thank you so much. So we'll bring Dr. D'Amore and Dr. Bahardi back into the conversation here a little bit, and we'll try to answer some more of these questions live. So let's see here. There was an additional question that I know we talked about briefly earlier about the letter difference in treatments having, 4-letter difference, 6-letter difference. And there's a question here, until what line is the 4-letter difference still reliable? And when you go over the 2060 or the 2070, how many letter difference is considered reliable?
Unfortunately, that's a clinical question, and I don't feel qualified to answer that. I don't know, Kapil, if you have better insight than I do.
Now,
I mean, they're talking about a chart when they're
yeah.
talking about letters, and when they say 3 lines, they're talking about 3 lines on a chart. But these nuanced questions, I don't have an answer to, unfortunately.
Well, we can table that one for now, since I do know that Dr. Zarbin had to pop off here. So, in any of these questions we don't get to in the chat, if you feel that you need more clarity or you feel you would like more elaboration, feel free to email us at info@fightingblindness.org, and we can take these questions offline as well. So, when it looks for some of these FDA approvals for, let's say, the stem cell treatment, what would we be looking at for a timeline for that? I know that a treatment has to go through all the phases before it goes up for that FDA approval, but is there an estimate of how long that process takes?
Yeah, Bailey, I think that's a good point. FDA is very mindful of that these technologies take time to develop. But because they're so promising, they do have also accelerated the process of approval. They have now faster approval for all of the stem cell-based therapies. They call it advanced therapy approval process, which allows the sponsor of the technology to actually get market approval or rolling-based market approval by phase 3 trial in many cases. Many of these are now in phase 2. So my hope is that in the next few years, as if they look promising, they might be reaching a stage that they would be getting FDA approval for commercial use.
Wonderful. Thank you for that. And Dr. Giamore, maybe this is something that kind of comes off of your presentation, is as we talk about wet AMD, dry AMD, and then progressing into GA, what does that cycle look like? Which one usually comes first? And if you have one, does that mean you are going to develop into one of the others?
I know Kapil's also welcome to jump in on this one. Um, I mean, what we know is that wet AMD is a lower proportion of all of the AMD that's diagnosed, but progresses faster and therefore causes more damage. Dry AMD progresses slowly generally, um, and can transition to wet. I don't know what the number is there. Is it like 10% or 20%, Bill?
Yeah, that's about
It's not a
10%.
high
Yeah,
percentage, but yeah.
10%.
So it can transition, but obviously not a straight line because it's a small percentage that transitions. People can have both at the same time, and that becomes very complicated. Some people say that when you clear up the wet, you can see dry RGA underneath it. I don't think, unless Kapil, you know more about that, I think that's not totally cleared up. Which one was there first?
Yeah.
What was that? I'm going to let you call the questions, Bailey, because there's a lot of them there, and I'll
There
let
Yeah.
you
are
figure out
a
which ones you
lot.
want answered.
Yes, there are a lot, and a lot of these have some similar topic, characteristics as well. There's a lot of questions that we've seen come through about ARIDS and ARIDS 2. So I wonder if you guys could talk just a little bit more around that, its efficacy, if it's something that if there's a difference between the ARIDS and then ARIDS 2.
I can start on that. ERITS was the first natural history study for AMD that was conducted by the National Eye Institute. And the more newer version of that is ERITS 2, which was done almost now already 10 years ago, I think. And that led to the supplement that is being widely used for delaying AMD symptoms. So that's the main difference between them. It's just a temporal thing.
Perfect. Dr. Diamore, anything that you would like to add on that topic?
No, I mean, I just think it's a very effective vitamin. It has antioxidants in it. A lot of systemic diseases are inflammatory. So I did see a question about COPD and AMD, and my first response was no. And I looked it up, and in fact, there is a correlation, and that goes back to the idea of this, which I'm sure people have heard about, systemic inflammation. I think the simplest thing to say there, for instance, is when you smoke, you cause systemic inflammation. That's the most obvious one. I don't know of a disease for which smoking is not a risk factor. So I think in some people's diets can be inflammatory. So, I mean, there is also a genetic component. So, for instance, if somebody has AMD in their family, they may want to get tested genetically, and if they don't want to get tested genetically, they could just start taking the vitamins. The vitamins are not going to hurt for sure. And, the earlier, in my opinion, that you start to address what the issues are, the better off you are. That's why I don't smoke, eat a Mediterranean diet. All those are easy to say, much less easy to implement, but taking vitamins, not so difficult. So, I think, if there's, any suggestion that there's AMD in the family, then taking them even before anybody says, oh, you might have drusen is not a bad idea.
Great point. Yes, those preventative measures that we can all take are definitely will help. So there's another question that came into the Q&A piece around stem cells. So can the stem cells be injected both intravitreal and subretinal?
Yeah, so short answer is yes, but the ones that are being injected in the intravitreal will definitely have a different mechanism of action as compared to the ones that are being injected subretinally. The ones that are being injected intravitreally will likely be anti-inflammatory, protective of protective of neurons and cells of the eye. They're not going to be restorative in nature, so they're not going to go in and replace damaged cells. Whereas once injected or transplanted in the subretinal space, are transplanted with the aim that they are restorative, so they will replace damaged RPE cells or damaged photoreceptors. And with that, the hope is that those cells will stay forever in patients' eyes. And the ones that are being injected intravitreally, they will probably die off in a few weeks. So patients may need multiple injections during their lifetime for those stem cells to keep working.
Wonderful. Thank you so much for that. And Louise, I want to bring you back into the conversation here a little bit. When you were sharing your Home Depot custom glasses that you've created to share with your friends and family what you see, it reminded me of, we get a lot of questions around, to those in the community of what is your visual acuity? How do you interact? And I think it's a true testament that low vision is a spectrum, that a lot of times it is not all or nothing. So aside from using that tool to share with your loved ones about your experience, is there anything else that you have found helpful when you are describing your experience, or how maybe you adapt certain skills and how you've interacted that way?
Yeah, I think there's when we talked about attitude, it's just, what was very frustrating for me, and my process was very accelerated over the last 6 years, is you have a very hard time transitioning when you have to try to do things, the same things you have done, and you try to do the same way you did before, and that does not work. So the most relevant thing in terms of attitude, you need to find different ways for doing things. Let me get very simple examples. For example, putting toothpaste in your toothbrush. So there's no way it's not gonna work. If you're gonna try to do the same way you did and looking at it, it's not gonna work. So you need to find a different way to do it, which is adapting to your new reality. Or if you wanted to cut your fingernails, there's no way you're gonna cut your fingernails the same way you did it before. There are other different devices or how you're going to approach that, but you need to find your own way to do it. Otherwise, it's not going to work. In my case, for example, I also lost the capacity to see color. So I see very little. Everything is black and white and shades of gray. And so you need to find your own way to how you separate your clothes, or if you don't want to have a problem, use everything black and white. And at the end of the day, I think the most important thing here is to find the way you want to do things, but in a different way. Don't try to force yourself to do the same way, because you're going to get frustrated. It's not going to work. I'm not saying I solved all my problems. I still have a lot of problems. I still get frustrated. I cry. I do everything that all of us know what it happens. But I think the most important is don't fight the old way. It's not going to work. You need to find a different way. So sometimes you need to sit and find how I'm going to solve this with my new reality. Um, on the other hand, I think I'm reading much more books, because audible is a wonderful thing, and that helps me a lot. And I'm doing different things that I cannot go outdoor and run on the street, because I just cannot do it. But do exercise in a different way. So I think there is an issue about altitude, and don't fight your old way of doing things. And that's probably the most relevant thing, because it only gonna get a little worse, and you're gonna get very frustrated. And that would be my advice.
Wonderful. Thank you so much for that information. Well, as we're getting ready to wrap up today, I did notice a lot of questions coming through the chat about Stargardt disease as well. So we will actually be planning to have a webinar directed about Stargardt in April. So stay tuned for more information about that. We will be able to dive a little bit deeper into some of that content as well. For those who maybe did not get their questions answered today, we can take those offline and try to get you some answers there. So like I said, feel free to reach out to us at our info@fightingblindness.org. But just to wrap us up today, I wanted to share with you an exciting information of an event that we have coming up in June in Fort Worth, Texas. It's our United Envisions conference. And this is going to be sharing more about cutting edge research and therapies. It's going to be an opportunity to network with individuals. We will have assistive technology booths there, and as well as many sessions who are led by fantastic clinicians and resources such as you received here today. So please join us in Fort Worth, Texas. And then I also wanted to once again give a special thank you to Johnson & Johnson for their sponsorship of this AMD webinar, as well as our many other national sponsors that we have. And then lastly, as we wrap up today, you will be receiving an email later on this afternoon with a survey, because we want to help understand the experiences of people living with geographic atrophy, including their treatment journeys, access to care, and informational needs. Your responses to this will help us be able to be better advocates and be able to more effectively advocate for those individuals living with GA in our community. All these responses will be anonymous, and this this survey will only take about 10 minutes to complete. So we'd greatly appreciate your feedback on that, so we can better understand the needs of our community members. But lastly, I want to just once again thank all of our fantastic speakers today for their contributions to this discussion. We covered a lot of ground around AMD today, so I hope you all enjoyed this session. If you have any questions, feel free to email us. And if not, have a great rest of your weekend and stay tuned for the recording in the upcoming week. Have a great day, everyone.